Hello, this is Dr. Rina Rani Ray. Today I'll be discussing on microbiology of beer. Beer is the most popular and most consumed alcoholic drink around the world. It has been reported that the Egyptians were the first to document the brewing process around 5000 BC. It is also believed that the first brewers were part of primitive cultures of Mesopotamia. The first beer was basically made from grain, water, and spontaneous fermentation due to wild yeast present in the air. The birth of modern beer was during the early Middle Ages when German monks introduced hop as a bittering and flavoring agent. With the arrival of the Industrial Revolution, beer production moved to mass manufacture, allowing beer to be produced on a much larger scale. Beer is produced commercially by the controlled fermentation of wort, a liquid rich in sugars, nitrogenous compounds, sulfur compounds, and trace elements extracted from malted barley. Fermentation is a process by which glucose is converted to ethanol and carbon dioxide. So it can be expressed in this formula where sugar, that is glucose, is converted to ethanol and carbon dioxide with the release of two molecules of ATP. Behind this simplified chemical reaction is a series of complex biochemical reactions. These reactions, known as glycolytic pathway or EMP pathway, involve a number of enzymes and the reactions take place anaerobically inside the cells of brewing yeast. Breweries carry out the fermentation by a continuous process in which the beer moves through a series of stirred vessels for a period of 40 to 120 hours. After the ethanol has formed, the beer is transferred to maturation vessel and the flavor is naturally refined. The alcohol in beer actually is produced by yeast fermentation. The final alcohol content is determined by comparing the initial and final specific gravities. Normal mixing and transferring the wort to a fermentation vessel will aerate the wort. Initially, the yeast will grow quickly as they use aerobic respiration. But when the oxygen is depleted, the yeast will switch to fermentation and produce ethyl alcohol. Carbonation is done to beer by bubbling in CO2 or by a secondary fermentation in which sugar or unfermented wort is added to the bottled beer. Commercial beer is aged several weeks and pasteurized in the bottle. Some beers are filtered rather than pasteurized to remove contaminating microbes. There are two types of beer fermentation. One is known as a ledger, another is a alley. Ledger, the beers made with yeast that settle on the bottom. Here, the yeast involved is Saccharomyces euverum, and here the yeast remains in the at the down of the can. And in LA, beers made with the yeast, that is the Saccharomyces cerevisiae, which floats on to the top of the brewing vats, resulting in a cloudier beer. They tend to have a higher alcohol content than the ledgers. So here we can see the top fermentation, that is the LA type, and the bottom fermentation, that is the ledger type. Here we can see in the top fermentation type, it requires warmer temperature, that is 65 to 75 degree Fahrenheit, whereas in the ledger type, a cooler temperature, that is 45 to 55 degree Fahrenheit is required. The raw material used for beer production are cereal, that is a barley malt, rice or maize, then hops, water and yeast. The malting process converts the starch in the cereal into fermentable sugar, which is extracted from the malt during mashing. Hops. What are hops? Hops are the flowers or cones of a plant called Humulus lupeus. They keep beer fresh for a long time and help to retain its head of foam, a key component of beer's aroma and flavor. Extracts from the hop are used as a preservative and to add bitterness to the sugar solution. And finally, the key role is played by the yeast. Main two yeasts are the Saccharomyces cerevisiae, that is the top fermenting one, and the Saccharomyces euverum, that is the bottom fermenting yeast. Yeast converts the sugars into alcohols during fermentation. In the absence of oxygen, we can see the pyruvate formed from glucose through glycolysis can follow two different pathways. And depending on the type of cell, it can be converted into ethanol or alcohol and carbon dioxide to the alcoholic fermentation pathway. Now, regarding the brewing process, beer, like any fermented food, is a microbial product. Microbial activity is involved in every step of its production, defining the many characteristics that contribute to its final quality. While fermentation of cereal extracts by Saccharomyces is the most important microbial process involved in brewing, a vast array of other microbes also affects the complete process. Barley is the source of the fermentable sugars in modern beer. Malt is formed by germinating or malting barley seeds. The plant cells convert starch in response to fermentable sugars. Additionally, plant proteases reduce the total protein and make amino acids available for the yeast. The malt is then dried to 50 degrees centigrade and roasted at around 75 degrees centigrade. So the first process of brewing involves the crushing of the malt seeds, then the filtration, then boiling with hops and sedimentation, then cooling, and finally the fermentation and bottling. So this is the main essence of brewing process. So first one is the milling the grain, where different types of malt are crushed together to break up the grain kernels in order to extract fermentable sugars to produce a milled product called grist. Then comes mash conversion. The grist is then transferred into a mash tun where it is mixed with heated water in a process called mash conversion. The conversion process uses natural enzymes in the malt to break the malt's starch down into the sugars. Now it is time for lautering. The mash is then pumped into the lauter tun where a sweet liquid known as wort is separated from the grain husks. Now it is time for boiling. The wort is then collected in a vessel called the kettle, where it is brought to controlled boil before the hops are added. Now here we can see that the hops are being added. After boiling, the wort is transferred into the whirlpool for the wort separation stage. During this stage, every malt or hop particles are removed to leave a liquid that is ready to be cooled and fermented. Now it is a time for fermentation. To start the fermentation, yeast is added during the filling of the vessel. Yeast converts the sugary wort into beer by producing alcohol, a wide range of flavors and carbon dioxide, which later the process it is used to give the beer its typical sparkle. After fermentation, the young green beer needs to be matured in order to allow both a full development of flavors and a smooth finish. After reaching its full potential, the beer is filtered, carbonated, and transferred to the bright beer tank where it goes through a cellaring process that takes 3 to 4 weeks to complete. Once completed, the beer is ready to be packaged and it is now uh, going on for bottling and for final marketing. So, some words about the health benefits of beer. Beer has some anti-cancerous properties. Hops used in beer, a flavonoid compound called xanthomyl plays a major role in the chemoprevention of cancer. It also shows reduced risk of cardiovascular diseases as beer contains vitamin B6, which protects heart diseases by preventing the buildup of a compound called homocysteine. Moderate beer consumption increases bone density, thereby preventing the risk of fractures and osteoporosis. Moderate beer consumption also shows some anti diabetic activity against type 2 diabetes. Beer is a good source of vitamin B12 and folic acid, which prevent anemia and is required for maintaining normal growth, good memory, and concentration. According to biomedicine, regular beer drinkers would have lower blood pressure compared to people that consume similar amounts of wine or other liquors. Beer increases the potency and impact of vitamin E, which is a major antioxidant in the body. It is important for maintenance of healthy skin while also showing down the aging process. Gallstones, regular consumption of moderate amounts of beer would affect the cholesterol levels and decreases bile concentration, leading to a reduced risk of developing gallstones. Beer consumption also boosts the level of good cholesterol by 10 to 20%, thus reducing the risk of dementia and cardiovascular diseases. And finally, beer possesses a number of digestive properties, which include the stimulation of gastrin and gastric acid, and thereby they helps in digestion. So, moderate consumption of beer can give a number of health benefits. So, this is all about the microbiology of beer production and their good effects. So, thank you.